This lesson's called Going Viral, not like viral videos on the internet. Today we will learn about viral indicators and reference viruses of contaminated waters. Last lesson we learned about harmful pathogens found in recreational waters. Since there are so many different types of pathogens and it's expensive to monitor for all of them, scientists use non-pathogenic indicator bacteria to measure if there has been a contamination event. Although indicator bacteria typically suggest that there are pathogens in the water, sometimes they do not correlate with the presence of viral pathogens, like norovirus. Viruses can be found in contaminated waters even if indicator bacteria concentrations are low. This has to do with the differences in their makeup, like their structure. In comparison to bacteria, viruses are often more resistant to wastewater treatment and can survive for a long time in the surface waters. For example, viruses are not as easily destroyed by chemicals like bleach and ammonia as bacteria are. Because of this, scientists have suggested also testing for viral indicators to make sure they do not miss these important pathogens when testing contaminated waters. That's right, viruses can be indicators and references of contamination too. A virus is an infective agent that typically consists of a nucleic acid molecule and a protein coat, is too small to be seen by light microscopy, and is able to multiply only within the living cells of a host. Although you're probably most familiar with viruses that infect humans, all life around you, including your pets and plants, can be infected by viruses. Another group of organisms that are constantly battling viral attacks are bacteria. Viruses that infect bacteria are called bacteriophage, or phage for short. These viruses are commonly found in our gut along with our healthy gut bacteria, and therefore are also found in our feces and in wastewater. These non-pathogenic viruses are considered viral indicators, meaning they do not harm us, but they can let scientists know that fecal contamination and thus potentially pathogenic viruses could be present. Coliphage, viruses that infect E. coli indicator bacteria, are one example of a viral indicator of fecal pollution that can easily be cultured in the lab. We learned last lesson that bacteria grow on agar plates filled with nutrients. Phage are grown in the lab by covering a plate with their favorite food, bacteria, and allowing them to infect for about 24 hours. The result is almost the opposite look of a bacteria plate. In this case, the bacteria form a cloudy lawn all over the plate except for areas where the bacteria have been killed by the coliphage, which appear as cleared spots called plaques. Each plaque is called a plaque-forming unit, or PFU, and originates from a single phage infection. This is similar to culturing enteric bacteria, where each bacteria colony forming unit, or CFU, is formed by a single bacterium replicating. Scientists can count these plaques to determine how many coliphage were present in the water sample. Coliphage are a great example of an easy-to-work-with virus, and the U.S. Environmental Protection Agency is currently working on developing recreational water quality criteria for coliphage that will hopefully improve how swimming waters are monitored in the U.S. There are some other viral indicators and reference viruses that are useful for understanding wastewater pollution in surface waters. For example, pepper mild mottle virus, or PMMOV, which you can learn more about by clicking the link in the description, is a plant-infecting virus that can be found in wastewater pollution around the world. Viruses like PMMOV are not as easily grown in the lab from water samples. So how do we test for those viruses in contaminated waters? Genetic material. Scientists use genetic material to find these viruses in contaminated waters. Each virus has either a DNA or RNA genome. In order to get the viral genetic material, first the viruses need to be collected from the contaminated water source. Virus concentration is crucial to being able to detect viruses in water. To do this, the collected waters can be filtered through a dead-end ultrafiltration system that catches the viruses and other pathogens on an ultrafilter. The pore sizes are so small that only water can pass. Additionally, pressure is needed to push the water through, so a pump is used to squeeze the water through the tubing and out of the filtered membrane. To keep the viruses fresh, the work does not end here. The filters are preserved in a dark cooler for a trip back to the lab. Note that this is only one example of how viruses and other microbes are collected. There are many methods used around the world. Scientists continue their work in the lab. Sometimes days can be 12 to 14 hours long, making sure everything is processed before the pathogens decay and the data are lost. First, all of the collected pathogens need to be removed from the filter. Oftentimes, an additional virus concentration process is used to further reduce the volume of the sample. Next, the genetic material from the virus needs to be extracted. While scientists use specialized kits in the lab, they are based on simple properties that you can replicate using items from your own kitchen. If you're interested in these steps, make sure you do the DNA extraction activity at the end of the video. Finally, since the scientists cannot see if the genetic material is present, a test called quantitative polymerase chain reaction, or qPCR for short, is done. qPCR targets one gene or area of the virus's genetic code and makes the same copy of that gene repeatedly, like a biological copy machine. Each copy of the gene is lit up by DNA-binding dyes, so the more copies of the gene present, the brighter the light, allowing the machine to count how many copies are present. This reaction happens exponentially over time, making even the smallest amount of genetic material measurable. Each sample is compared to a quality control positive to figure out exactly how much of the particular virus's DNA or RNA is present in the environmental sample. Usually, one copy is equivalent to one virus. DNA and RNA extractions, similar to the ones used to identify viral indicators, are used in your everyday life. For example, when you get sick, the doctor needs to test if you're sick with a virus like SARS-CoV-2, or if it's something else. To do this, they need a sample of body fluids, like a mouth or a nose swab. This swab is taken to the lab where the genetic material is extracted and a genetic test is completed. Another way you may use DNA in your life is with a family ancestry test. You start the process of the DNA extraction by spinning in a tube and adding buffers to send it off to the company that does the rest. Your DNA is used to match which regions your ancestors are from. Want to do your own DNA extraction at home? Did you know that every living organism has DNA, including your pets and plants? You can try this at home with fresh strawberries and a couple of other ingredients found in your cabinets. Strawberries have 8 times the amount of DNA per cell compared to human cells, so it'll be a lot easier to see their DNA than our own. Try this activity yourself or watch the video by following the link in the description. I hope you give the DNA extraction a try at home. It's really cool to see the cloud of genetic material. Scientists use the same steps, just with fancier chemicals and filters to get the job done. So if you like the activity, you might like working in a molecular biology lab like us. This video was narrated and created by Mackenzie Kerr with support of Dr. Maya Breitbart and Dr. Aaron Simmons. This work was supported by the following grants from the U.S. National Science Foundation. Any opinions, findings, and conclusions or recommendations expressed in this material are those of the authors and do not necessarily reflect the views of the U.S. National Science Foundation. Thanks for watching. Check out more resources in the description to learn more about this project and to see other videos.